Hi friends, today I am here with you to discuss that of the important points from the chapter genetics, and it is the part 2 section, the continuous section of the part 1. I am already discussed that of the important points from the genetics in the part 1 section. Please watch that, and it is part 2 I am doing here. So let's start. So first we are discussing about the experiments, that is the experiments which will be mentioned in that of the NCERT textbook. So first is about the Griffith experiment transformation principle. So transformation principle, he done his experiment transformation principle in 1928. Year should be memorized. 1928. It is done by who? The Griffith. And this experiment, transformation principle experiment, get repeated by McCarthy, MacLeod and Avery. So this experiment is repeated. For more results, they repeated the experiment. And who repeated that? It is McCarthy, MacLeod and Avery. Okay, then the discovery of nuclein. The discovery of nuclein. I am discussing some more important points in this section from that of the chapter genetics. So here in genetics the important word is nuclein, and this is proposed by Dr. Frederick Mishker. Dr. Frederick Mishker. Clear. Then the nuclein get changed to that of the nucleic acid. This name was later known as to be the nucleic acid, and it is done by who? Altman. So nucleic acid. The name get changed to nucleic acid. It was originally nuclein, and this was done by who? The Altman. Clear. Next is about X-ray diffraction. The X-ray diffraction, which was really helpful in that of the DNA structure. So X-ray diffraction is by Rosalind Franklin and Dr. Wilkins. Rosalind Franklin and Dr. Wilkins. Then this X-ray diffraction is really helpful for the DNA structure, for the identification or proof of a DNA structure, and this DNA structure was done by Dr. James Watson and Francis Crick. It is almost all students are get studied in that topic about in the previous classes. So about DNA structure. DNA structure, it was explained by that of the Dr. James Watson and Francis Crick. Then the semi-conservative nature of DNA. The semi-conservative nature. Okay. It will be detailed discussed in that of the chapter genetics, that I continued that section. Okay, please watch that. And here I am discussing about only the term semi-conservative. The semi-conservative nature of DNA get proved by who? Yeah, it is by Watson and Crick. The same persons. Next is about VNTR. Okay, in the last section of the genetics 2, okay, molecular basis of inheritance, the +2 second genetics chapter detail discussed about that of the satellites, that is mini satellites, VNTR. So VNTR varied number of tandem repeats. That is the expansion for the VNTR, and this VNTR is example for what? Mini satellite. Okay. So VNTR is an example for what? Mini satellite. And here it is long repetitive sequence. Mini satellite is long repetitive sequence. Then example for short repetitive sequence is junk DNA. So next is about what? Junk DNA. And this junk DNA is an example for what? Short repetitive sequence. It is long repetitive sequence example VNTR and short repetitive sequence example junk DNA. Okay. Next is about tRNA. tRNA. Yeah, tRNA, which is the soluble RNA. Okay, that should be memorized. That point should be memorized. Which is soluble RNA? The soluble RNA is tRNA. In NCERT, there is a structure which is given for that of the tRNA in the 2D and 3D shape, inverted L shape like structure. So soluble DNA, soluble RNA, it is what? tRNA. Okay, then they are discussing about some RNA polymerase 1, RNA polymerase 2, RNA polymerase 3, and that indicates what? Okay, so first is about RNA polymerase 1. I'm shortly written polymerase. Okay, so RNA polymerase 1 indicates what? rRNA. Then RNA polymerase 2 indicates what? hnRNA. Okay. Then next is about RNA polymerase 3. So RNA polymerase 3, which is tRNA. Okay. This tRNA is soluble RNA. Okay. Then this tRNA has, I'm already told that it has two type of structure, that is 2D and 3D. And 2D shape of tRNA is what? Clover leaf model. Okay, clover leaf model. It is 2D, and 3D is what? Inverted L shape. Inverted L shape. These structures are given in that of the NCERT textbook. So go through that. Okay, then it will be clear for you. Okay, then next is about what? Yeah, ZYA, that is lock apron. Okay, there is lac operon. In lac operon, I'm detailed discuss this lac operon in the next section in that chapter video. It is here I'm only discussing the important points about this lac operon. When we looking this lac operon, we come across with that of the three enzymes. So which are they? SED. SED indicates what? SED produce beta galactosidase. Beta galactosidase. And then Y for permease. And the last one, that is A. It is for transacetylase. Transacetylase. Clear? Then next about DNA fingerprinting. Okay, DNA fingerprinting was proposed by who? Alec Jeffrey. Alec Jeffrey. Okay. Then some point wise topic from that of the NCERT line. Go through that lines, then you can come across these points, that such as when chromosome number 1, when we consider about the chromosome number 1, it has 2968. Clear? Then when we considered about that of the Y gene, it is only have 231, that is it is having the few. And next is about VNTR. Yes, this example for mini satellite already discussed, varied number of tandem repeats, and its size it is 0.1 to 20 kb. Clear? 0.1 to 20 kb. Okay. Then the chromosome 1 is the last discovered one while we considering it. So the chromosome 1 is the last completed, and it completed in that of the May 2006. Okay, May 2006 last completed, which is that chromosome number 1. Chromosome number 1. Then next is about automated DNA sequence. Automated DNA sequence, and it is by Frederick Sanger. Okay, Frederick Sanger. Then one example for that of the free-living non-pathogenic nematode. These are some points which should be memorized for that of the need. Okay. The next point is about the free-living non-pathogenic nematode. Free-living non-pathogenic nematode, which is that, yeah, Cynor habitis elegans. Cynor habitis elegans. Then the last point in this video, it is about Human Genome Project. What is the short form of this? Yeah, HGP, and it is 13-year project. 13-year project, and the year is 1900 to 2003. 13-year project. And here the contribution for this project is given by that of the two companies. It is US Department of Energy and the National Institution of Health. Okay. So please go through these points, then it will be very helpful for you in NEET syllabus. Okay, thank you very much.